My first task today is to introduce Dr. Brian Ballios. Dr. Ballios comes to us from Toronto, from the world-renowned Stem Cell Research Lab led by Dr. Vanderkooy, funded in part by the Foundation Fighting Blindness. And so this is one of those labs doing translational research that is supported by the Foundation. Dr. Ballios has a PhD in stem cell biology and in regenerative medicine from the University of Toronto. And this was initiated through an MD PhD program. And what that is Brian made a huge commitment to do both a medical doctorate and a doctorate of philosophy or a PhD at the same time. He started his med school, he left, he did his PhD, he's come back to medical school with hopes of taking some of that research forward. His doctoral studies were focused on the design and of new approaches for the transplantation of stem cells in the treatment of retinal degenerative diseases. As a matter of fact, there will be some time left aside at the end of his talk for questions. Please hold your questions till he's had a chance to do his entire talk. And at the end, what we will do is we'll ask that, as best you possibly can, raise your hand. Someone will come around. There'll be microphones, and we'll try to keep it as orderly as possible. Without further ado, I'd like to introduce Dr. Brian Ballios, and he's going to introduce Vision Research today. Brian?
Well, thank you very much for having me this morning. It's a great pleasure to be here. Thank you very much to the organizers also for inviting me. The title of my talk is Vision Research Today, but I'd also like to maybe give you a window into vision therapies of tomorrow. As was previously mentioned, I did my PhD work in the laboratory of Professor Derek Vanderkooy at the University of Toronto. And there I was exposed to much of the exciting progress towards the cures for blinding eye disease. And it was inspiring mentors like Dr. Vanderkooy and other scientists that are being funded by foundations like the Foundation Fighting Blindness that are really putting forward the search and pioneering the quest for the next generation of treatments for vision loss. Now, for the most part this morning, I'm going to be talking about diseases that affect the light-sensitive tissue at the back of the eye, that is, the retina. And I'll be talking about therapies which may have an impact on a number of different diseases, including those that affect the very young, like retinitis pigmentosa or Leber's congenital amaurosis. These are conditions which many of you are familiar with and some of you may be living with today. And also conditions that affect adults in middle age, like diabetic retinopathy, and age-related macular degeneration as we move into our more senior years. But what all of these conditions have in common is that they cause irreversible vision loss, and that many of our current therapies today, that is to say our drug therapies, can only serve to stave off the progression of that disease, but they don't always represent a restorative approach to vision therapy, giving back sight that has been lost. Drug therapy, or as often called pharmacotherapy, they both mean the same thing, can support the normal functioning of the visual cells that remain during degeneration. Examples of these drugs include some that you may be familiar with, like Lucentis, a once-a-month injection for age-related macular degeneration that stops many of the biological processes that are occurring in the retina, like the new blood vessel growth, which can injure the retina over the course of the disease. And organizations like the Foundation Fighting Blindness fund many of these exciting avenues of research, and I'm sure you're going to hear more about that later today. And there are many more drugs still in the basic stages and early stages of animal research. But these drugs are unlikely to correct many of the root causes of the disease, which often lie in the realm of the cells and their specific genes that are dysfunctional. These drugs, however, may prove to be very important as adjuvant therapies or additional therapies when combined with future vision-preserving or vision-restoring therapies. Now, one of the benefits of drug therapies is that the approval process for these therapies, as composed to some of the others that I'll be talking about a little later, is often more rapid. These therapies don't often make permanent changes to the cells in your body or the genetic code when they're applied, and they're often derivatives of other drugs that are already being used elsewhere in the body. So some of those regulatory benchmarks are already out of the way. In fact, the Foundation Fighting Blindness is funding at least 4 different research teams that are looking to develop drugs now here in Canada to protect and preserve the retina. An example of this is Professor Rod Bremner at the Toronto Western Hospital, who's been developing drugs to prolong the life of those light-sensitive cells in the back of the eye, the photoreceptors, for people with diseases like retinitis pigmentosa and other inherited eye disease. He's also been developing drugs that can delay the growth of retinoblastoma tumor, which is a blinding eye cancer that occurs in very young children, and neuroprotective drugs that may have benefit for different types of eye diseases. I'd like to speak now, though, about 3 potential approaches that may serve to restore vision that has already been lost. These fall in the realm of gene therapy, retinal prostheses, and stem cells and cell therapy. And you're going to hear a little bit more on each of these topics later today in the breakout sessions. The first area in gene therapy, really, to begin to explain this area, I need to begin by explaining what a gene is. And a gene essentially serves as the basic blueprint for how a cell functions. It tells a cell in our body what to do. Our genes tell our muscle cells to be muscle cells, they tell our brain cells or our neurons to be brain cells, and they tell the cells in our eyes, like our photoreceptors that respond to light, to continue to do so. And in the last 20 years, our understanding of the genetic basis for many eye diseases has exploded. There's now over 200 different genes that have been identified to contribute in some way to blinding eye disease. And this expansion in our knowledge of the genetic basis of disease can be seen as both a challenge and an opportunity. It's a challenge because it highlights the incredible heterogeneity, the incredible variety that makes up the cause of human blindness, and that it's going to require concerted efforts from multiple experts in the field. And it's going to require some considerable funding from multiple agencies like the Foundation Fighting Blindness and the National Health Research Programs to help researchers understand how these genes contribute to disease. But it represents an incredible opportunity because for the first time, we're able to identify the molecular underpinnings of disease. And it gives us hope that at least at first, for diseases that are affected by single genes, they may be amenable to the techniques of gene therapy. Now, what is gene therapy? Well, gene therapy is replacing those genes that are lost or damaged and that cause retinal disease. The tissue, the retina at the back of the eye, is particularly amenable to approaches like gene therapy because it's so accessible. It's one of the only nervous system tissues that we can see from the outside of the body. Gene therapy may involve the use of drugs to increase the function of genes that are defective, but in many cases it means replacing that damaged or defective gene. And replacing a gene is a complicated process, but it's best served by delivering that gene to the cells in the eye in a virus. And many of us think of viruses as being bad things, being agents that cause disease and cause illness. But in the case of gene replacement therapy, viruses carry good genes, or genes that actually work, to the cells that need them in the eye. And they replace those lost or damaged genes. And early treatment with gene therapy may prevent vision loss and may actually allow cells that no longer function properly to restore their function so that they can properly transmit light to the brain. Gene therapy assumes that those cells that are essential for vision in the retina are still surviving and remain alive. And there's a great deal of research here in Canada, in the US, the UK, and internationally on these potential therapies. And many of you may have heard of some of these. And these include replacement of single gene defects. For example, in diseases like Leber's congenital amaurosis, there's been trials into the replacement of a gene called RPE65. This is a single gene that, when dysfunctional, can cause blinding eye disease from a very early age. And early clinical trials have shown that patients have some improved vision with gene replacement therapy when delivered in a virus. There are Canadian trials of gene replacement therapy going on right now, funded in part by the Foundation Fighting Blindness. For example, a trial led by Dr. Ian MacDonald at the University of Alberta for the gene replacement therapy of choroideremia. But what do you do for people who've completely lost their vision, where the cells that normally work in the retina to transmit light information to the brain are lost or destroyed? Well, the next two therapies that I'm going to talk about have some potential hope for that. Retinal prostheses is something you may hear a little bit more about today. What these are, essentially, are electronic devices. You can imagine them, in effect, as a digital camera that is linked in some way to the same visual pathways that transmit light information to the brain. An example is the Argus II. It's a product that's recently been approved in the US and hopefully soon in Canada. It consists essentially of a camera placed in a pair of eyeglasses, very similar, if many of you have seen the Google glasses that are out right now, that sends electronic signals to a chip that's implanted in the retina. This is a very small chip, about 3 millimeters by 3 millimeters square. And that chip stimulates the same pathways as if light were falling directly on to a normal functioning retina. Now, I have to say that at this stage, the sorts of visual images that are coming from these devices are still fairly rudimentary. They can return some black and white vision, and many patients are unable to see very fine details, but they can see things like lights on and off and hand waving and very large block text. So it is a very important advance for those patients who are beginning with no vision at all. But a very large question in vision research is there a way that we can give back biologic sight to the human retina? Is there a way to restore the damaged tissue and help the retina heal itself so that it can function in a way that nature intended it to? And this is really the goal of stem cells and cell therapy. And any talk on stem cells should begin with a definition of what is a stem cell. Well, first of all, a stem cell is a Canadian discovery. And in the late 60s and early 70s, Dr. Till and McCullough at the University of Toronto described cells isolated from our own bodies that are able to exhibit the two cardinal properties of stem cells. The first is that as they divide, they make more copies of themselves, a process we call self-renewal. And their second property, and perhaps the most inspiring and encouraging for future vision therapy, is that these stem cells can be coaxed to turn into any type of cell in our body. This includes muscle cells, brain cells, and retinal cells. Now, stem cells come in many different flavors, if you will, many different types, and many of you have probably heard of some of these types, like embryonic stem cells or adult stem cells, which are isolated directly from tissue. And two strategies I'd like to talk about using these stem cells are the first around stem cell transplantation, and that is isolating these stem cells in a dish in the lab, coaxing them to self-renew, to proliferate and grow, and also to transform into the types of cells that we need for vision therapy. A second strategy, which I'll touch on a bit later, is encouraging those stem cells, when they're resident in the tissue, in the eye, to begin to grow and transform into the types of cells we need so that effectively our eye tissue heals itself from the inside. Now, many of you may think that stem cells sounds a bit like science fiction. It's a bit far off, but I have to say that there are some very early clinical trials already underway employing stem cells to replace damaged cell types in the retina. In particular, researchers have already begun RPE transplantation, and RPE is the retinal pigment epithelium, and that RPE is a layer of pigmented cells at the back of the eye. These pigmented cells help keep light that enters our eyes around for the retina to process that light information and transmit it to the brain. Now, this RPE can be derived from multiple different types of stem cell sources, but most importantly, researchers have seen early functional improvement in preclinical studies in both rats and monkeys, and already in the US, a company called Advanced Cell Technologies has begun a phase 1 clinical trial using embryonic stem cell-derived RPE for the treatment of diseases like Stargardt's macular dystrophy and dry age-related macular degeneration. And clinical trials are also planned in the UK under Pfizer to begin later this year, and trials in Japan for early in 2014. So clinical trials in stem cell research have already begun, and they begin to help us understand the regulatory pathways from moving cell-based therapies to the clinic. This has already been worked out in most part for drug therapies, but there are different hurdles to be overcome with cell therapies, especially around proving that these transplants will be functional after they're delivered to the eye. Now that's retinal pigment epithelium transplantation. But really the only option to restore the lost vision cells, the photoreceptors that are lost, would be photoreceptor transplantation. And stem cells are a good source of these photoreceptors. The RPE supports those photoreceptors in their survival, but it's really the photoreceptors that do the heavy lifting. They transform the light signal into the electrical signal that's sent to the visual centers of our brain. And again, there's multiple sources for photoreceptor cells, both from embryonic stem cells and different types of tissue-specific stem cells. And a number of groups in the US and the UK have been reporting, and many of you have probably heard about in the news, functional improvement in mice after transplanting these photoreceptors into the eyes. So where do we go from here with stem cell therapy? Well, I mentioned before that there are multiple sources for stem cells, and different sources have different issues around them, in particular ethical issues around the tissue sources. And so the big question we have to ask ourselves is, what is the best cells for cell therapy? And I would argue that those stem cells that are already resident in our adult tissues may be the best ones for therapy. They would avoid many of the ethical concerns around using embryonic or fetal tissue. They may allow for patient-specific harvesting. You could imagine isolating some of these stem cells from your own eyes in a small biopsy to be grown in a lab to derive cells that are specific, your cells, specific to your body, that can be transplanted back into your eyes. And the fact that these stem cells are already present in our tissue means that we have that option to potentially stimulate those cells to help the retina heal itself from the inside. In fact, in the eye, there's a population of retinal stem cells that you can see. And if you take the opportunity to look into the eyes of your neighbor, you would see that the dark band around the outside of the eye that sits between the colored part, the iris, and the white part of the eye, that 1 in 500 of the cells in that darkly pigmented band is a retinal stem cell. Now, these cells help to build the retina when it's growing, when we're in the womb, and after we're born, when we're very young. It helps to grow that retina, but those cells stop dividing when we become an adult. And there's many evolutionary reasons why that might be true, and I won't go into all of them, but suffice it to say that we don't want these cells to continue to proliferate in the adult because our eyes have finish growing to the size they're intended to be, and generally nature tries to keep cells in our adult bodies from dividing too much to prevent the growth of tumors. Well, Dr. van der Kooi and his colleagues back in 2000 hypothesized that maybe there were stem cell populations in the margin of the eye, and they were inspired by the fact that cold-blooded vertebrates like goldfish and zebrafish continue to grow their eyes throughout adult life. Their stem cells that build their retinas never stop proliferating. So we hypothesized that if we could take these stem cells out of the eye and find the right factors to get them to grow, we could get the human retinal stem cells to begin to proliferate in a dish. And in fact, we were able to do that. And we were also able to show that these stem cells have the potential to be transformed into any type of cell in the retina. We've characterized, at this point, very well their potential to turn into multiple different cell types. What we need to do now is harness that power to turn them into one particular cell type. And this is where I came in with my research during my PhD, which was focused on new approaches to coax the stem cells into turning into specific types of cells, in particular the photoreceptors, the heavy lifters for vision in the retina, and also around new ways to transplant those cells back, to get them to integrate into a diseased retina so that they can make a functional connection and restore some sight. And transplantation, I feel, will likely be the first approach used for any stem cell therapy in the retina, far before trying to coax the stem cells already resident in the eye to begin to rebuild it from the inside. But there are still major hurdles to be overcome. Hurdles around what particular combination of chemical factors and how do we coax the stem cells to turn into photoreceptors? How do we ensure that those cells are distributed in the tissue after we deliver them? How do we keep those cells alive after we deliver them? And how do we ensure that those cells are making the proper and appropriate connections with the retina? Well, with the generous help of Foundation Fighting Blindness funding and other sources of funding from the federal government, we've shown the potential functional benefit of these cells in restoring visual function in mice. And I showed through my work that we could isolate combinations of chemical factors to derive photoreceptors from our stem cells with over 90% efficiency. This gives us a great starting point to begin to use these cells in transplantation. I've also looked to combine that delivery with using bioengineering and biomaterials, injectable gels that can carry these cells to the eye, right to the site where the normal photoreceptors are degenerating, and support their survival while they take time to integrate and form connections. I've shown that these gels can both support the survival and the integration of these cells, and in the last 1.5 to 2 years, our mice are beginning to show the early indicators that these cells are functioning normally and integrating into their eyes. I'll stress again, though, that the ultimate dream of stem cell therapy is to encourage those stem cells that we know are already present in the eye to heal the eye from the inside, very similar to the way that when we get a skin wound or a skin cut, our skin can heal itself, encouraging the stem cells in the eye, which are normally quiet, to rebuild the retina after injury, either from genetic dysfunction or from acquired injury, is really, I think, the future direction for stem cell therapy. So in conclusion, Canadian scientists like Professor van der Kooi, myself, and the others you'll hear today are really trying to push forward an international effort to move towards sustainable and regenerative approaches to vision therapy. And this is thanks to the generous support of foundations like the Foundation Fighting Blindness. And I really see today as an opportunity for you to learn more about our research. And I encourage you to be bold in asking your questions. Scientists like myself really enjoy these events. We find inspiration in your journey, and we share your eagerness for answers. So I thank you all for coming today, and I hope you find it informative and inspiring. Thank you very much. I'm happy to take any questions. I'm being told we might have time for about 2 questions, so please. So the question was, has there been any success in using stem cells for the regeneration of optic nerve? That's an excellent question. The optic nerve really represents the pathway that cells use to signal from the retina to the brain. The cells that actually make up that pathway reside within the retina itself. And there are researchers who are looking to replace these cells, but the major challenge is getting them to extend small processes all the way down the optic nerve and to the brain. And there's a number of different approaches that are being used, but most of that work is still in the very basic stages of research. And there are no current clinical trials underway, to my knowledge, to regenerate those pathways in the optic nerve. And that's going to require a really concerted effort from people not just in the field of vision research, but also people in brain research to understand how cells form these very long connections to the brain. So that's an excellent question. And I think a major difference between transplanting cells to restore the pathways in the optic nerve and the transplantation of photoreceptors is just the distance that these cells need to make a connection. With the photoreceptors, it's a short jump for them to make a connection to the next cell in the chain in the retina. But for those cells that make up the optic nerve, they're stretching their what we call axons, their processes, all the way from the retina at the front to the back of the brain, a very long distance by comparison. So it's an active area of research, and I think there's been a number of encouraging early studies, but most of these are in the preclinical stage at this point. So we look forward to seeing where those directions go, where they take us. Thank you. Yes, so there's a number of approaches that are looking at eliminating barriers to cell transplantation in the retina. Scar tissue is one barrier that can often serve to limit how well cells that we transplant integrate into the retina. So there's a number of different chemicals that researchers are using and still in early animal models to try to remove scar tissue to allow cells to integrate. That's an excellent question. It adds an extra layer of complexity to our studies. So nothing yet in clinical trials for that, but a number of promising preclinical work that we can digest that scar away, remove it from the eye, and put our cells back. Thank you very much.